All right, it looks like we are live. So, hello everyone, and welcome to this month's Rocky Rails discussion. Actually, we will have something new. We actually have two speakers. We're going to present the work that they're going to present the work that they have been doing together. And yeah, as a reminder of the netiquette, please use your full name. Keep muted during the talk, because there will be plenty of time to ask questions at the end. And when you would like to ask a question at the end, you can raise your hand and show yourself on the video when you're speaking. But yeah, without further ado, I am going to stop sharing my screen and I will give over to our first speakers, Mike Wong.
Hi, everyone. It's so good to be here at the Rocky World Seminars series. Let me go ahead and share my screen here and go full screen. If at any time something breaks, you stop hearing me, the slides aren't advancing, somebody just yell at me. I know Zoom sometimes has a mind of its own. So I'm Mike Wong. I am a NASA Sagan Fellow at the Carnegie Institution for Sciences Earth and Planets Laboratory in Washington, D.C. And I'll be co-presenting today with my good friend and colleague, Dr. Anirudh Prabhu, who's a research scientist and PI at the same lab, Carnegie Sciences Earth and Planets Laboratory. So I'm a planetary scientist and astrobiologist by training, and Anirudh is a data scientist with expertise in developing and applying AI and machine learning to applications in all kinds of geochemical systems. And today we're going to be talking about some really exciting research of ours at the forefront of astrobiology on searching for life in the solar system using machine learning and pyrolysis GCMS. I'll give the first half of the talk talking about our biosignature detection technique, but motivating that with biosignature detection in general. I'll introduce the instrumentation and data collection for our technique, and then Anirudh will take it from there and talk about the data processing pipeline, our machine learning methods, the results, interpretation, and our future work. So let me begin by giving you a taste of how astrobiologists think about life detection. I was on the scientific organizing committee for the Biosignature Standards of Evidence Community Workshop held in the summer of 2021. This was a joint effort between NASA's Nexus for Life Detection, or NFOLD, and NASA's Nexus for Exoplanet System Science, or NEXUS. These are two of the research coordination networks under the greater umbrella of NASA astrobiology. So the big idea was we should come together as a community and seek agreement on how to identify, interpret, and report on potential biosignatures. And so the attendees created a white paper that was open for community input and is now published on the archive. It's 86 pages long, which is quite a bit. So in case you don't want to read all of it, I've got a single slide summarizing it for you. So the framework for the standards of evidence for life detection follows 5 key questions. They are as follows. Number 1, have you detected an authentic signal? 2, have you adequately identified the source of that signal? 3, are there abiotic sources of that signal? 4, is that signal consistent with the expressions of life? And 5, are there independent lines of evidence that help you raise or lower your credence in having detected alien life? And because all these questions are interrelated, we've grouped them schematically like this. Number 1 and 2 feed back upon one another to understand the detection. Have you actually found something of interest? And 3, 4, and 5 do the same to understand our interpretation. So have you understood, for instance, the environmental context in which you have found that particular signal? Now, 5 is really critical. Every strong scientific theory, as we know from the scientific method that we learned in school, should make new predictions that can be tested. And so through question 5, this becomes an iterative process. And although they're numbered 1 through 5, they're not necessarily done in order. For instance, a modeler might start with asking questions 3 and 4, so that once observationalists or experimentalists go and do 1 and 2, we'll be able to interpret the meaning of those signals. So let's look at a classic case study to see this framework in action. And let's review the case for and against the detection of alien life in the Martian meteorite ALH 84001. So this is the famous worm image produced via scanning electron microscopy, which was, oh, that looks like really interesting microscopic form, potentially a fossilized alien life. So that's number 1. Looks like we may have found some kind of fossilized microbial life. In addition to those morphologies, you also find complex organic material in this meteorite, in particular polycyclic aromatic hydrocarbons. Also, if you look at the carbonate globules in this meteorite, they're enriched in carbon-13 to an extent that on Earth could indicate biogenic activity. And then finally, this Martian meteorite has these curious magnetite crystals with distribution and shape similar to those produced by magnetotactic bacteria on Earth. However, further study revealed that these microscopic forms are about an order of magnitude too small and have no internal structure, so they may not actually be indicative of cellular life as we know it. That complex organic material, well, it litters the cosmos and may be contamination from the meteorite having spent the past 13,000 years on planet Earth. Those carbonate globules enriched in carbon-13 are also known to form abiotically now, and those magnetite crystals could have formed by shock alteration of the meteorite. So how does this all stack up against our 5 questions from before? Well, okay, number 1, have we detected an authentic signal? Yes, we use standard electron microscopy and chemical analyses. Those seem to be robust. We know we are looking at something real. Have we identified the signal? We know the sources. We know that this comes from a certain imbalance in carbon-13 versus carbon-12. We know that this is indicative of a particular kind of organic molecule, for instance. But what we've found is that a lot of these signals can be produced abiotically, specifically that's carbon-13 enrichment in the magnetite grains. And is the signal consistent with expressions of life? Well, actually not, because the cell structures look like they are too small to have been viable cells. And then finally, are there independent lines of evidence? Well, you have to look at the context of this meteorite, having spent the past many thousands of years sitting on planet Earth, could have been subject to a lot of contamination. Let's do one more example because I know there are a lot of exoplanet fans in the audience. The example of dimethyl sulfide on K2-18b. This was a gas that was reportedly detected in the atmosphere of K2-18b in James Webb Space Telescope data. And this gas is primarily produced by microbial life on Earth. And the discovery was responsible for a whole lot of press coverage a few months ago, including this New York Times headline that read, "Astronomers detect a possible signature of life on a distant planet." So have we done it? Have we witnessed the first discovery of life beyond our solar system? Well, the case is perhaps not so clear-cut, and the biosignature standards of evidence questions show us exactly why. So as this study has been continuously reanalyzed, the data has been continuously reanalyzed, we found that other analyses conclude that actually there is no statistical evidence for spectral features in that data set. If there are those spectral features in the data set, other studies have shown that perhaps molecules with methyl groups other than DMS provide an equally good fit to the data. So the identification of the signal is in question as well. Are there abiotic sources of the signal? Well, this molecule dimethyl sulfide is known to exist on comets and in the interstellar medium. And is the signal consistent with expressions of life? I'd have to say we don't know yet. Biochemists and astrobiologists would have to think very hard about why this particular molecule would be produced by metabolisms on K2-18b, which is purported to be a Haitian world. But at the same time, there are questions about whether or not K2-18b is indeed a Haitian world, and some studies suggest that it could be consistent with a lava world as well, which means that would any expression on a lava world be consistent with life as we know it? And then, are there independent lines of evidence? A recent study showed that perhaps there are other complex molecules in this atmosphere, like diethyl sulfide and methyl acrylonitrile, which could, further advance this framework for understanding what is happening in this planet's atmosphere. So this back and forth that's occurred just in the past few months is indicative of, just how long it will take, years, if not decades, for the scientific community to come to some kind of consensus regarding exoplanet biosignatures. But still, it's an extraordinarily exciting time. With JWST, we can finally ask a specific astrobiological question about a specific gas in a specific exoplanet's atmosphere. And that's cool because we've never been able to do that before. Can we answer all those questions yet? As you see in this slide, not yet, but there's still so much incredible work left to be done. So with that recap of past and present biosignature assessment, let's now turn our gaze toward the future and also return to our solar system. So we're embarking on potentially a golden age of astrobiology in our solar system, with numerous missions being explicitly tasked with trying to find evidence of habitability and life in our cosmic backyard. So we're actively trying to return samples from the surface of Mars. The Europa Clipper mission is on its way to the Jovian system to get there in 2030. In the 2030s, the Dragonfly mission will visit Saturn's moon Titan, and the latest decadal survey recommended an Enceladus flagship level mission to go and search for life in that moon's icy plumes. So now is the time to try to develop cutting-edge in situ biosignature techniques for solar system exploration, techniques that will be more effective than the ones that we've so far tried. So what makes a strong biosignature technique? Well, all biosignature techniques, I think, should fulfill four main criteria. The first is that it should be robust, which means immune as possible to false positives and false negatives. Second, it should be agnostic, which means capable of identifying life, even if that life has non-earthly biochemistry. Third, it should be probabilistic, which means we should be able to quantify our confidence in having detected or not detected alien life. And finally, it should be flight ready. It should rely on instrumentation that NASA, ESA, or some other space agency is actually going to strap onto the back of a rocket and launch into space. So Anirudh will talk about the robustness and the probabilistic nature of our biosignature technique. I'll leave that for him to speak about when he looks into the machine learning pipeline and our results. I'll mainly be focusing on motivating the agnostic nature and the flight readiness of our technique. So let's start with agnostic biosignatures. What is an agnostic biosignature? Well, as you all probably know, a biosignature is a feature that indicates or suggests the presence of life in some environment. And we tack on this word agnostic to say that this biosignature should not be specific to life as we know it on Earth. It should be capable of detecting unfamiliar life forms as well. And so this is because there's a growing recognition in the astrobiology community that we cannot expect life in the universe at large to have the same specific details, especially chemical details, as life on Earth. So we need to do some abstraction away from those certain details that may be universal to life on Earth, but may not be fundamental to life everywhere in the universe. One way to do this is by appealing to some metric of complexity. Life is, after all, the quintessential complex evolving system, and complexity is maximized somewhere between total orderliness and total chaos. And so one might see life as the system in which information structures flows of matter and energy, and that structuring leads to complexity that can be the foundation for developing agnostic biosignatures to search for life in the universe. Now, before we dive into our own proposal for an agnostic biosignature, let me quickly recap a few examples of agnostic biosignatures that other labs have developed in recent years. The first is chemometric fingerprinting, developed by Georgetown University professor Sarah Stewart-Johnson and her colleagues. The idea here is basically to use DNA strands as different colored sticky notes, and the more complex your sample is, the more different DNA strands you'd expect to bind to it. So on the bottom, you see a simple ordered sample, a crystal, a non-living sample, and it only interacts with a few number of different DNA strands. But a more complex sample, like the cell wall of an alien life form, will interact with all kinds of DNA. So here we're measuring the complexity of our sample by the diversity of DNA sequences that that sample interacts with. And for more information on this, please check out Sarah Stewart-Johnson's 2018 paper published in Astrobiology. Another strategy is the molecular assembly index. This was pioneered by Glasgow University professor Lee Cronin and ASU professor Sarah Walker and their colleagues. So this basically asks, how many unique transformations must I make to construct an object of interest, whether that is a series of building blocks, letters that form a word, or chemical reactions that make a molecule. So using the assembly index, you're basically measuring the complexity by the number of unique steps that it takes to construct a molecule. And I like to think of this as the Lego block principle. If you get a Lego set from the store and your instruction booklet is very, very thin, then you know it's an easy set. But if the instruction booklet looks more like a dictionary, then you know you're going to put a lot of time, energy, and information processing into making the final product. And the interesting thing is, it seems like life is the only process capable of creating high molecular assembly index molecules. So in this slide, I'm showing you 4 different molecules: asparagine, tryptophan, penicillin, and ATP. And all 4 are used in life, but tryptophan and asparagine can be made geochemically, whereas penicillin and ATP, well, their only known production mechanisms are life. And so there seems to be some cutoff, perhaps around molecular assembly index 15, where above that, the only process that can make those molecules is life, and below that, life or non-life could be responsible. So the molecular assembly index, combined with the copy number, how many of those molecules you find in a certain sample, is said to give you a measure of the information content of the system in question. And for more on assembly theory, please see the growing body of literature by Walker, Cronin, and colleagues. So while molecular assembly index characterizes the complexity of individual molecules, the technique that Anirudh and I are working on is complementary. It's based on the molecular distributions of all the molecules in a particular sample. Now, this idea has been discussed in the literature for the past couple of decades, but the idea is basically this. We might not expect living samples and non-living samples to have we would expect them to have different distributions of organic molecules. So in this graphic, what I'm showing you here in the gray is an abiological sample. This corresponds to a distribution of some regular thermodynamic distribution. And in red, you see a biological sample corresponding to the molecules that are specifically found in life. Now, there's no guarantee that alien biochemistries will use the same biomolecules as life on Earth. So the distribution of organic molecules in alien life might be very different from the distribution of molecules in life as we know it on Earth, but they will still be very different from the distribution of molecules in abiological samples. So fundamentally, why is it the case that the distribution of organic molecules in biotic samples should be different from those in abiotic samples? Well, for this, allow me to make an analogy to statistical mechanics. So say you have a hot cup of tea. The positions and the momenta of all the particles in that teacup constitute that teacup's microstate. And it just so happens that there is vastly many more microstates corresponding to a cold cup of tea in a room temperature room than correspond to a hot cup of tea in a room temperature room. And assuming that all these microstates are equally likely to be visited by this ensemble of particles, this explains why hot cups of tea tend to cool down over time. This is our second law of thermodynamics. This is our arrow of time. The analogy that I want to make to life is this. There may be a similar high-dimensional phase space, not of positions and momenta of particles, but of molecular patterns that differentiates life from non-life. Now, the curious thing about life is that lifeless chemistry becomes self-organized into biochemistry. Non-living worlds transform into living worlds in the opposite direction from a warm cup of tea cooling down. It seems to defy the second law of thermodynamics, but of course, you have to remember 2 important things about life. Number 1 is life is an open system, which means that it can dissipate free energy to raise the entropy of its environment and maintain its own local order, and that life grows in complexity over time due to selection pressures from its environment, in particular selection for functions like homeostasis and resiliency. So we explored this idea of selection for function in a paper that I led titled On the Roles of Function and Selection in Evolving Systems. It's a paper that was written by a vast group of scientists of very different backgrounds, as well as philosophers of science. And I encourage you to look at this paper for more of our thoughts on selection for function and how that relates to evolution, information, and biosignatures. But I won't say any more about it here because we want to get back to our biosignature detection method. So the technique that Anirudh and I are about to introduce to you has its legacy in the work of Gutenberg et al. 2021, which was published in Life. And in this study, they used electrospray ionization Fourier transform ion cyclotron resonance mass spectrometry, or ESIFT-ICRMS, to examine the distributions of organic molecules in a diverse array of samples, like living samples, and you see E. coli on the right, non-living samples like the Murchison meteorite, a carbonaceous chondrite you see in the center panel, as well as origin of life experiments, the outputs of origin of life experiments like the famous Miller-Urey experiments that you see on the other end. And so they trained a multi-class logistic regression classifier and achieved a 92% accuracy in differentiating between living samples and non-living samples. And this is really great. This was exciting, except for one thing, and that is the Applicability to an astrobiology mission. You see, ESIFTICRMS is not just a super complicated name, it's also a super complicated chemical analytic technique. It's something that will probably not fly on a mission in the near-term future. So in our work, we seek to rectify that. And here finally is the paper that Anirudh and I are proud to present to you, a robust agnostic biosignature based on machine learning and pyrolysis GCMS. The lead author is our good friend and colleague, Jim Cleaves. The team also includes Greta Heistad, who's a mathematician at Purdue, George Cody, an organic geochemist at Carnegie, Sophia Economon, who's a graduate student at Johns Hopkins University in the Earth and Planetary Science Department, and Bob Hazen, a mineralogist and astrobiologist at Carnegie. So, say you want to go to Europa and find evidence of alien life. How would you do it? You'd probably send a spacecraft to land on the surface of this icy moon, taste this plume detritus, this material that likely originated in the habitable subsurface ocean below. And on your spacecraft, you might fly a pyrolysis gas chromatograph mass spectrometer. Why? Because these instruments are flown to interplanetary destinations all the time. And if you're not familiar with what a pyrolysis gas chromatograph mass spectrometer is, don't worry, just think of it as a very fancy oven that not only bakes your cake, but tastes it for you too. In more detail, here's how it works. There are four main steps: flash pyrolysis, followed by chromatographic separation, electron ionization, and then finally analysis by quadrupole mass spectrometry. So what flash pyrolysis does is basically flash heating your sample to over 600 degrees Celsius. This volatilizes all the organic matter in your sample, creating these pyrolysates. And the pyrolysates then flow through a long, thin chromatographic column that is coated in phenolpolymethylsiloxane. This basically helps us segregate the different molecules from our sample based on their chemical nature. Of the molecules that exit the chromatographic column at any given time, they can be further segregated by mass after electron ionization, which allows them to be selected by the quadrupole mass spectrometer. What are the advantages of pyrolysis GCMS? Why is this an instrument that can and is flown to interplanetary destinations? Well, the advantages are many. First, the flash pyrolysis requires very little sample prep, no fancy derivatization needed. You just heat up your sample and you're ready to go. The gas chromatograph is much simpler and more reliable than methods that depend on liquid mobile phases, which may not work so well in the low pressure, low temperature environments native to many interplanetary destinations. The 70 electron volt electron ionization is a relatively low energy technique, which is good if your spacecraft is on an energy budget, as most of them are. And then the quadrupole mass spectrometer provides ample information about the composition of our sample and is a mainstay of planetary missions. So the data that our pyrolysis GCMS gathers looks like this. You get a forest of peaks in 3 dimensions. The mass to charge ratio is on the x-axis, the retention time in the gas chromatograph is on the y-axis, and the intensity, or the number of those molecules that we're counting per second, is on the z-axis. And so if you found something that looked like this on Europa, you would be absolutely ecstatic, because this is what the average Earth bacterium looks like to a pyrolysis GCMS. Now, the thing is, we're not guaranteed that life on Europa is going to look exactly like life on Earth, right? It may use a slightly different set of organic molecules, so it would be potentially foolish to go and try to detect life by saying, oh, I'm just going to look for one of those peaks there, or one of those molecular fragments. Instead, there may be something about, again, the overall distribution of the organic molecules in our samples that help us identify samples that are living versus samples that were never alive. And so what we've been doing at Carnegie for the past several years is taking all sorts of organic rich samples, things that are alive right now, like bacteria and plant matter, things that used to be alive, but over millions or even billions of years have been degraded into petroleum, coal, oil, and shale, and things that have never been alive, but still contain copious organic matter, like the contents of carbonaceous chondrites, and again, the results of those chemical synthesis experiments, like the Miller-Urey experiment. Now, hopefully, just by eye, you can appreciate that the distributions of organic molecules in all 4 of these sample types are different from one another. And the question that faced us after we noticed that was, can we quantify their similarities and their differences from one another and train a machine learning algorithm to tell if something was once alive or not? And the answer to that, excitingly, is yes. And I'll turn it over to my colleague Anirudh Prabhu to explain to you how.
Thanks, Mike. I'm going to share my screen as well really quickly. Let me know if everyone can see my slides and hear me clearly. Perfect. Thumbs up. Great. Okay. So, hello, everyone. My name is Anirudh Prabhu. I'm a research scientist and PI at Carnegie Institution for Science. And today I'll be taking you through the machine learning component of one of our recent big advances where we developed a machine learning method to distinguish between biotic and abiotic samples. And let's see if the slides actually proceed. There we go. So to start off, as Mike mentioned, I'm going to reiterate the point of what makes a good biosignature detection method. A good biosignature detection method should meet four main criteria. It should be agnostic. That is, it should be capable of distinguishing signs of life different from Earth's known biology. It should be probabilistic. That is, the model or method developed must be able to quantify the confidence in the detected biosignature. The method must be robust. That is, it should be consistent and accurate at detecting biosignatures and be immune to a certain extent to false positives and false negatives, right? And it should also be flight ready. Now, Mike talked about what makes a biosignature detection method agnostic and flight ready. As I go through this talk, I'm going to address how our method is probabilistic and robust. So to start off, Mike already introduced the PyGCMS machine. We used the PyGCMS machine to analyze about 134 samples in our first iteration. So the 2023 paper that we are actually talking about in detail today used these 134 samples. We've since analyzed about 408 or 410 samples. And those papers will be out very soon. So I'm very excited about that as well. And these samples that we've analyzed are a good mix of biotic and abiotic samples, including leaves, fossils, meteorite, nylon, and a lot more. And as Mike mentioned, the data produced by the pyrolysis GCMS has three main attributes. So we're looking at a 3D matrix-like structure where we have rows represented by the scan numbers, which is the proxy for retention time, the columns represented by the master charge ratio, and intensity values for each row and column. And as Mike mentioned, most of the times researchers using PyGCMS identify specific peaks and use those peaks to identify specific molecules. The identified molecules are used to ascertain the nature of a sample, but that's not what we do. We look at the entire data set. We look at the positions of the peaks, we look at the intensity values, we look at the relationships between the peaks themselves. And for example, you can see the aggregated data as Mike presented for bacteria, plants, petroleum, and meteorites. And by looking at the entire landscape that is emerging, our model is able to find recurring patterns in the data, and we use them to predict the biogenicity of a sample. So to get started, this is an ultra simplified workflow of our method, which starts with running the PyGCMS on a bunch of samples. We next pre-process the data to get it ready for our machine learning models. These steps include smoothing of the mass spec data, baseline removal, normalizing the intensity, peak detection, and lastly, flattening the mass spec data and binning it to make it more computationally efficient for our models. That's because each of our samples contains about 500,000 data points in some sense. So we really need to make this a little bit more computationally efficient, especially because we want a lightweight model when it's going to be flown to some other planet. So once the pre-processing is done, we could get to the crux of our approach, which was building and testing many different classifiers that could predict whether a sample was biotic or abiotic. We trained quite a few classifiers, including support vector machines, extreme gradient boost, and neural networks. But what we found was that our random forest model was the one that performed the best. Now, as an aside, random forest is a very popular nonlinear method. It aggregates over many decision trees to improve its predictive capability. And in the upcoming slides, I'll go through the step-by-step process of how this model works. But to start off, when we had our base model uh, using the random forest method, we parallelly tested it and improved its generalizability using feature selection and nested cross-validation. And I'm going to say our current final model, so this was the last published random forest model, is able to distinguish between abiotic and biotic samples with close to 90% accuracy. A little bit of a spoiler, our current models are able to do that with many more samples as well, but that paper will be out very soon because we just submitted it yesterday. So now to explain the random forest method itself. We start by taking a random subset of the data. So we'll do a random 80-20 split of sorts, and we use it to build a decision tree. This decision tree classifier tries to predict whether a given sample is biotic or abiotic based on certain important features of the data that it observes, right? Now we keep building many such decision trees on different random subsets of our data. So we do this 500 times or 1,000 times. So when you have so many different decision trees, it produces a forest, right? Now that we have such a decision forest, we can predict the biogenicity of any new unknown sample. So what happens when we feed a new sample into the PyGCMS, create this spectra, and then try to understand what that sample actually is? We then put that sample's data through every single decision tree. We aggregate the results of the entire decision forest, and the aggregated voting results from all of these trees is then compiled to form a final prediction. And that's how random forest works. Now, because each tree was made from a random subset of samples, we can calculate what's called the out-of-bag error through the remaining samples and thus assess the prediction accuracy of our model. It's this error that we're trying to minimize through fine-tuning of the hyperparameters and feature selection. Now, how did we ensure that our model was robust and accurate? First, we divided our data into training and test data. We used a 70-30 split. 30% of the data was never introduced to the model until the final set of predictions was made. Now, with 70% of the training data, we ran nested cross-validation and found that the model maintained the same level of accuracy as our first set of improved models. We also ran this step many times using a Monte Carlo method to ensure the robustness and reproducibility of our model. Finally, we tested on 30% of that untouched data that I talked about and found that our model was still performing close to 90% accuracy on the test samples, thus showing the generalizability of our model. Now, once we had our model and predictions, next we wanted to explore and interpret our results. So what you see on the screen is a fairly simple PCA plot showing how similar or dissimilar our analyzed samples are. The first two principal components account for about 55% of the variance of the data. The other 18 plus components contribute to the rest of the 45% of the data, showing you how truly complex this data is. So we colored these samples by their biotic or abiotic labels. Well, at least we did so initially until we noticed that there was a clear bifurcation in the biotic samples that are starting from the left and spreading to the right. So as you can see, the abiotic samples are all clumped in the bottom left in orange, but the biotic samples are proceeding in two different directions. So we examined these two groups that emerged that we didn't initially train our model to actually distinguish. And we found that they corresponded to fresh biotic samples, so things that are modern biotic, recently been alive, and degraded biotic samples, or things that have been taphonomically preserved, like fossils or petroleum, things like that. So another point that I'd like to mention is reiterate the agnostic nature that Mike already talked about. So let's say in this scenario that you have 3 new samples you want to test using our method, right? You can see based on the molecular distributions produced by the sample, they'll assume their place somewhere in the PCA plot. Now, in this example, you can see that unknown samples 1 and 3 place themselves squarely amongst the existing samples. So sample 3 is probably going to be rightly predicted as a fresh biotic sample. But what about sample 2? Sample 2 behaves unlike any of the biotic or abiotic samples that we've trained on. In this case, it could mean one of two things. The first, the more obvious thing, is that it means that our test set can and should be expanded. As I mentioned, we are already doing that right now. The latest version of our paper, which we just submitted, was trained on 406 samples, and we're constantly growing our analysis pipeline, and we're growing the number of samples that we have. But the second more exciting possibility here is that the sample's molecular distributions are truly unlike any of the biotic or abiotic samples on Earth. So we've just identified an anomalous sample. This sample should obviously then be further studied to see whether it has potential for us finding life on other planets different from what we know on Earth, right? So to summarize this section of the talk, we developed a potentially agnostic method to distinguish between biotic and abiotic samples. This distinction is not based on the identity of any specific molecule, but rather on the molecular distributions themselves, which may point to a more fundamental difference between abiotic organic mixtures and biotic molecular suits. Lastly, our method is extendable. This method and many machine learning methods can be extended to analyze many different samples, including those from PyGCMS instruments that are used on board current space missions or planned space missions. So I've already talked you through just now about another reason our method is agnostic, but let me summarize the key points for a good biosignature detection technique once more. So a machine learning method like random forest can provide a quantified confidence for every single prediction that it makes. This is why our method is probabilistic. With our steps taken to analyze and assess the generalizability using cross-validation and our resultant model still maintaining close to 90% accuracy, I'm going to consider our method pretty robust at detecting biosignatures. And as Mike and I have already talked about, this method is flight ready. PyGCMS is one of those instruments that's constantly flown on space missions. So what are the active next steps that we're working on? So broadly, we're pursuing two big directions for our project. First is an astrobiological path forward, a lot of which I've talked about in this presentation. But the second is a paleobiological one. And both of these papers have recently been written up and submitted. So hopefully in a few months, you'll be able to read those results. On the astrobiological side, we have the opportunity to detect life on other planets when our method is combined with the flight-ready instrument like PyGCMS. And one of our other ongoing explorations is studying the data produced by a mixture of biotic and abiotic samples. Now, this is done to simulate real samples that we may find on other planets, because if there's a rover on another planet and it's picking up a sample to analyze, it's going to be really hard to clean the sample or process it in any way. So this is what we did, right? So to simulate and test this scenario, our intern, Emmy Slaughter, spent the last summer running a mixture of biotic and abiotic samples at various different ratios. So starting from 90-10 biotic to all the way 10% biotic, right? And we're really pushing the boundaries on this kind of mixture-based approach to see how much of an abiotic mixture can be in a biotic sample for us to still accurately recognize that the sample has some biotic components to it. I don't have too much time. I don't know. Yeah, I'm pretty much running out of time. So I don't have too much time to get into the results of the corresponding machine learning model. But suffice it to say that we're pretty much able to detect biotic components of a heterogeneous sample at small concentrations, as low as 10%. Now, don't hesitate to reach out to Mike or myself if you have any questions about this particular offshoot, but keep a watch for our paper, which should be out in a few months. On the paleobiological side, we plan to develop a more granular classifier. We've already done this with about 9 categories at this stage. We've received close to 300 samples from 25 of the world's leading paleobiologists. And we hope that our study on some of these ancient samples helps us improve our understanding of early life, including understanding the nature of some of Earth's oldest and enigmatic samples, like apex shirt. And we can hopefully discern their true nature. So in the last slide, let me also address one of the common concerns that we get studying paleobiological samples, studying really old samples. This is how we think about it. If you've been looking at science news over the last year, I'm sure you've seen some articles about a student who recently identified and translated an extremely old and degraded Roman scroll that was carbonized by Mount Vesuvius in 79 CE. The machine learning method was able to detect and identify characters from a very, very degraded scrolls when experts couldn't read anything from it. This is how we're thinking about the paleobiological samples. In spite of the degradation on very old samples, there's still some information left in the molecular distributions for us to make predictions about them. And we hope that our method can be used to study such samples as well. So that's about it from my side. I'd like to end this talk by thanking everyone for their attention and for our funding agencies for making this work possible. That's it. Let me know if you have any questions.
Perfect. Thank you so much, Anirudh and Mike, for your amazing talk. I'm really looking forward to results of upcoming space missions. If we can actually find some biosignatures, will be amazing. So yeah, if anyone has any questions, please raise your hand and show your video when you're asking the question. Does anyone have any questions? Anjali?
Well, I have so many questions. That was amazing. Thank you, both of you. Which one do I pick first? Okay, I guess I had a question for you, Anirudh, about you were mentioning how you flatten the data before putting it in the machine learning. I was wondering if you could talk a bit more about that. And I guess presumably you're taking the bits that are most informative, I'm guessing, and out of the bits that you have to flatten, I guess out of convenience to make this sort of feasible, if in the future there was if we could fly bigger equipment and stuff into space, like, is there something hidden in that flattened information that could help even more in the future, maybe?
That's a really good question. And let me actually share my screen again. So the flattening is pretty the flattening was not necessarily done for the computational feasibility. So let me just show this aspect. So what we had was a bunch of matrices, right? And so each table was meant to be fed in as is to the machine learning model. But when I flatten it into a single very large table, it reads as a sequence of events, which it makes it a sequence of like characters, which makes it more useful and applicable to most machine learning models. Think about everything that reads your text, right? Like a chat GPT. They prefer this sort of single long table, and the flattening was done mainly for that. The binning that happens after the flattening was done for the computational feasibility, right? So I have this as a backup slide mainly because I had presented this to some of our students and interns on what they should be doing. And I wrote here, well, it's ready for ML now that you've flattened it, if you have enough processing power. But really, no one does at this stage, right? So we kind of found the optimal way to bin our data for that computational complexity. I don't think I would change the flattening step if we have more like computational power, or if we have the ability to fly more complex machines. If we are able to create, and this is something that I'm sure myself and a lot of other researchers are working on, we're able to create a machine learning model that very fundamentally uses this 3D structures. I'd like to flatten them, stop flattening them for that. And that kind of structure works on video data and image data, which has a little bit of a different, I'm going to say, data structure and distribution than tabular data, time series data, because these are all spectra, right? So I'm treating them as 2 time series going off in different directions. So that's a really good question. And I think the binning is the key part of reducing the complexity, is my short answer to you. And that's what would change the moment we have the ability to fly something better and better computational hardware down here.
Awesome, thank you.
Any further questions? Because I have one myself, a more general and perhaps slightly more controversial question. One of the main properties that you indicated that a good biosignature should have is being agnostic to life, that it's not specific for life on Earth. But I just kind of wonder, like, would there be any merit in developing a few complementary biosignatures that are more specific for life as we understand it on Earth? Because this is the kind of life that we understand the best. So maybe we can develop more specific and perhaps more robust biosignatures for life specifically like we find on Earth to complement your more agnostic biosignatures. Do you think there's any merit in this kind of approach or not really?
Yes, absolutely. I think there is plenty of merit in developing biosignatures for looking for life here on Earth, or rather life like ours on Earth. And one of the reasons why is because we then gain enhanced capabilities for doing, detection of life here on Earth, searching for, different life forms right here. But a lot of that, has already been done in the literature. And so one thing that we are trying to do with our technique, as well as the rest of the community developing agnostic biosignatures, is to help widen our scope for detecting life elsewhere. And so, if there is life as we know it, say on Mars, and we bring back samples and then we sequence their DNA and we find that we can sequence, there is DNA to be sequenced, I think that would be exciting and a very robust detection of some kind of life on Mars. But there is, I think, no guarantee that life that is indigenous to Mars, that originated there and followed a completely different evolutionary story from life on Earth, would necessarily use DNA. So in the case that there is life on Mars that is different from us, sending a DNA sequencer to Mars or bringing back samples to Mars and then trying to sequence its DNA may come up completely empty. And in that case, we'd be missing life hiding in plain sight just because it doesn't use the same sort of genetic polymers as life on Earth. So that's the project that we're doing. In addition, our agnostic biosignature detection technique is also capable of identifying life as we know it. In fact, it does so quite robustly. And so hopefully the method that we're developing in conglomeration with all of the different methods that have been generated in the astrobiology community will allow us to attack the biosignature detection problem using multiple lines of evidence. And I think that's always the way to go, and to think about a pluralistic approach to detecting alien life. The more independent lines of evidence, the more independent lines of chemical analyses and techniques and interpretation that you can bring to bear, the more you can raise or lower your credence in having detected alien life.
All right, thank you. Then Joanna, go ahead.
Yeah, thanks, Mike and Anirudh. I've heard you talk about this before, but it's always cool to hear about. Anyway, I have also like kind of a broader general question. So as I'm sure you're aware, there's been a lot, I mean, lately, but just in like overall in the news about AI and like how that is impacting science or will in the future. And so as folks who are using this in a very like productive, I think benign way, right? I'm curious about kind of your general perspectives on that, and to ask a more specific question, like moving forward, what do you see as the essential role of human scientists in sort of biosignature detection versus AI broadly defined?
Yeah, I'll take that question to start, and then I'd love to hear Mike's perspective on this as well. So I think I've always been the strongest proponents of humans in the loop in any AI application, because I think it is extremely important all the way through, but at the very beginning and at the very end, with the curation of the data, making sure what's going into the training is good, is reliable, has good data quality, all these aspects. Because machine learning or pretty much any AI method does follow a bit of a garbage in, garbage out kind of scenario, right? And the second key aspect for humans to be involved in right at the end is the interpretation, right? Just because a model gives you some predictions doesn't mean that it's good. it is good in certain contexts. So I think not only does it require humans in the loop, you require at least a data scientist, at least someone who knows the data really well from the domain, both present during the interpretation to do this. So that's my first answer to your question is, I think that's never going to go away. But the role on some of the preliminary data collection or some of the preliminary predictions that are put out might have to be reduced in certain cases in astrobiological explorations because of sheer logistics of the further away you are from your own planet, the more time it takes to get data. So if I'm, say, on a rover really, really far away, still in our solar system, but really far away, and humans need to sit and make decisions about where to go explore, that's going to take a lot more time. And so some preliminary decisions can be made, still vetted by scientists, still vetted by, actual humans, but a little more autonomy may have to be granted because of sheer logistics. Now, I am also very aware that it's a little bit of the wild, wild west out there with AI development. And it's been honestly going on since 2020, right? And so what I think is needed generally, not just for science, it's very important for science, but for humanity in general on this hype surrounding AI technologies, is a little bit of regulations, right? On what type of development is happening, how can one actually use it? What are the potential ethical, legal, other concerns that may be coming from applications that they intend to build, but also unintended uses of potential technology. And that conversation is moving along, but not at the pace that the innovation is happening. So I understand that mismatch, and I, for one, would fully support. And we are also thinking about that kind of angle when we're doing this. So it is a necessary conversation to have, and I'm glad you brought this up, Joanna.
Yeah. As always, brilliant answer, Anirudh. My philosophy on AI is that it is a cultural technology, that will change the way that we do a lot of things, the way that the printing press did, or the internet, or cell phones. But as always, it will require, as Anirudh so wonderfully put it, humans in the loop. And one great example of that was in our recent runs of samples. We had over 400 samples now. And the AI model kept on making this one mistake about a particular thing called quisquite. And this is, some ancient rock. And and it kept on misclassifying it. And because it misclassifies quisquite and is learning about it, it's also misclassifying other things. And it took us looking at the data, looking at the results of the AI, and then iterating between myself, Anirudh, and Bob Hazen to say, oh, let's go back and look at the spectrum from quisquite. Well, it's very, very strange. It's almost like it's flat. It's almost like maybe the sample fell out of the sample tube. That's my hypothesis for what literally happened there. And so we have to reload the sample, rerun the thing, redo the... That is all in the human domain, right? That interpretation, that understanding, that physicality of things, saying what could have gone wrong here is that perhaps the thing fell out of the sample tube and we have to go and redo it, And so AI for me enables for the first time this extraordinarily multidimensional pattern search, right? Because we can only think in at most 3 dimensions, or at least I can. Maybe you can think in a few more, but AI can think in hundreds of thousands of dimensions at once. And that's critical for this kind of project where we have hundreds of thousands of different potential organic molecules in any given sample. Using AI, we're able to find those patterns very quickly, but also AI is only going to find the patterns in the things that it receives from us. And we're an essential step, as in the example from the quisquite thing.
Yeah, and I want to add another example to this if people don't have questions. Otherwise, I'll stop and wait. Because similar to this, there was another very interesting phenomena, and that basically shows some of what we know and some of what we don't know, even as scientists, right? So another sample that was constantly being predicted wrong by our model, this was in our test set, was the wasp's nest, right? And so we had assigned a wasp's nest as, oh, it's something that the wasp builds. We don't know what it's exactly made out of, and it's probably not plant material, right? So one of the tests we're doing is a wasp's nest photosynthetic or not? And We said, no, it's not photosynthetic. And it kept telling us that it's photosynthetic. So we're like, why is that? Like, either there's something wrong here, like Mike said, with the sample, or we're going to really need to check into it. So I think collectively, we just went into the literature to find stuff about wasps and their nests that we did not know, and found that actually it kind of is photosynthetic, because it builds it out of, it chews up bark and spits it out and builds like nests from that. And so it does come from plant material. So it's indirectly photosynthetic. So the molecular distributions are able to identify that, even if we didn't originally assign it correctly. And so there are absolutely times where we need to like check into it and fix ourselves or fix it. And both of these wouldn't be done without humans in the loop. So I think this is really helpful. Yeah. Anjali?
Yeah, I'm loving hearing about the variety of samples that you've all tried, like the wasp nests and the different kind of minerals and stuff. Going forward, are there sort of regions of parameter space in samples that you haven't explored that yet, but that you would like to go into? One of the ones I was thinking about is like, how much of a living thing do you need to be recognized as abiotic? Like if you have like just a DNA strand, is that enough versus like parts of a cell versus a whole cell and things like that, or totally different kind of samples?
Yeah, so the sample is actually quite small. It's just a couple tens or hundreds of micrograms of samples needed to run the instrument. Some sample types that we would love to get more of include fossilized animals and plants. Those are rare, but through Bob Hazen, he has a ton of geobiological colleagues that we can lean on to get samples from. And so we're working on curating a larger collection of those ancient biotic samples. Also, it's hard to find purely abiotic things here on Earth, right? Because life has taken over completely. So we do need to shore up and enhance the amount of things that we know are definitely abiotic. And that's actually going to be part of our project with this summer intern. M is here on the call, so M is going to help be helping us to run a suite of synthetic chemical samples as well to enhance the amount of abiotic samples that we have. And then, of course, you know, those wonderful samples from Mars. Like, could we please have Mars sample return? Please fund it. Yeah.
Very cool.
All right. There might be time for one small question if anyone has anything left. If not, then I think we're going to end it here for today. So thank you so much both for this amazing talk and amazing discussion. And yeah, we will be back next month, 3rd of July, with Remo Burr. So please attend there as well. And yeah, enjoy the rest of your day.
Thanks, everyone. Bye.
Thanks, everyone. Thanks, Mike and Anirudh. It was a great talk.
Thanks. Bye.